A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Chief Operating Officer of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members, as well as the broader scientific community, about trends relating to the commercial development of antibody therapeutics. Our expert speaker, Dr. Silvia Crescioli, will discuss the trends for the early stage clinical pipeline. Please note the webinar is being recorded. Please do add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I'll now turn the show over to our speaker.
Thank you so much, Jan, for the introduction. So today I'll talk about trends in the commercial development of antibody therapeutics with a focus of the early stage clinical pipeline. I'll start with the basics. I'll provide definitions, data sources, and our objectives in collecting this data. I'll then discuss trends in first-in-human studies for antibody therapeutics. And then I'll move to the main topic of our presentation, which is the current early-stage pipeline. I'll discuss about antibodies for cancer and non-cancer indications, and I'll provide an overview on formats, targets, and mechanism of action of these molecules. And then I'll conclude with the overall trends in the clinical pipeline. And I provide insight in trends in first marketing approvals and success rates. Definitions. I'd like to provide some definition because this gives me the opportunity to talk to you about our inclusion and exclusion criteria. Specifically to us, an antibody therapeutic is a recombinant protein-based molecule which has at least one antigen binding site that is derived from an antibody gene and that is evaluated as a therapeutic. So this excludes all the polyclonal antibodies from a natural source, antibody encoding DNA because it's not a protein, Fc only Fc fusion proteins because they only have the Fc portion of the antibody and not an antigen binding site, and everything that is just meant for diagnostics. Also, we are interested in commercial development, so we are interested in molecules developed by public or private for-profit entities. And we are interested in innovation, so we only consider molecules which are unique in composition of matter. So this excludes all the biosimilars. And also, in our analysis, we count each molecule only once. So by clinical status, we mean the most advanced clinical study that molecule has reached. And for each molecule, we will talk about first-in-human studies and first approval, which excludes all the second and third, etc. Sources of data. We rely on public disclosures from primary sources, which are mainly companies, company press releases, presentations, meeting abstracts, articles, annual reports, and also what the companies upload on their clinical trial registries, such as clinicaltrial.gov and others, and also on reliable sources such as regulatory agency documents and the WHO INN lists. We do not rely on secondary sources such as commercial databases, and this is because our inclusion and exclusion criteria is quite specialized, and they often lag in data update, and they might contain errors due to data processing. This is the reason why we have built our own database. So we collect this huge amount of data because our aim is to determine trends in antibody therapeutic development over time. And this allows us to analyze the data and prepare presentations like the one I'm giving to you today. And also we aim to determine clinical success rates for antibody therapeutics development as conducted by the biopharmaceutical industry. And also we aim to assess innovation in the biopharmaceutical industry. So, trends in first-in-human studies. This graph illustrates the number of antibodies entering first clinical studies each year. If you look at the green dotted lines, which refer to all antibodies, we can clearly see that from 2014, there is a steady increase in the number of antibodies which entered first clinical studies. If you look at the red line and the blue lines, referring to antibodies for cancer indications and for non-cancer indications, respectively, from 2016, the two lines, the two curves start to diverge, and from 2017, each year we have the number of antibodies for cancer indication that is almost twice the number of those for non-cancer indication. And now I will move to the main topic of the presentation, which is our analysis on the early stage clinical pipeline. I'll start with the status of these antibodies, and with this pie chart, I want to first show you the number of molecules we are talking about. As you can expect from the trends I just showed you, we have a huge amount of molecules. As of October the 2nd, we had 1,122 molecules in early stage pipeline. Half of them in phase 1, 20% in phase 1-2 and phase 2, and 10% at IND level, which means that the IND has been filed or approved, but the actual clinical trial, phase 1 or phase 1-2, hasn't started yet. We stratified our molecules according to the therapeutic area in antibodies for cancer and non-cancer indications. And as you can expect from the trends I just showed you, especially those of the recent few years, the number of antibodies for cancer indications is 2 times that for non-cancer indication. Also of note here, we have 4 molecules for which we couldn't assign a therapeutic area, and this is because these are molecules at IND stage, and sometimes companies do not release the therapeutic area at this stage. So we had to exclude them from our analysis. If you look at the status in these 2 groups, we can clearly notice that there is a difference in the proportion of molecules in phase 1-2 and phase 2. In the cancer group, we have more molecules in phase 1-2, 25% versus 9% in the non-cancer group, and 12% of molecules in phase 2 versus 35% in the non-cancer group. And these suggest that molecules for cancer indications are more likely to directly enter phase 1-2 study and assess both safety and efficacy compared to those for non-cancer indications. With this slide, I want to show you innovation. And this is our analysis on the proportion of unique targets in the 2 groups, antibodies for cancer indications and for non-cancer indications. By unique targets, we mean targets towards which there are no antibodies in late-stage regulatory review or approved. And interestingly, the proportion is very similar in cancer and non-cancer group, and is 59-60%, which means that the biopharmaceutical industry is engaging in innovative R&D. And now I'll focus on antibodies for cancer indications. We have stratified the molecules according to their format. Most of them are monospecifics, but now 1/3 of them are multispecifics. And then we have an 8% of immunoconjugates, which can be monospecific or multispecific, but we kept them as a separate category. And now I will provide you some information of these three groups, starting from the monospecifics. Here I'm providing an overview on the targets. I'm showing you a bar chart with the top 20 targets in terms of frequency. With the arrows, I'm indicating the unique targets. And I would like to spend a few words on the most frequent target, which is Claudin 18.2. And according to our definition of unique target, is not unique. But this is just because there is one antibody in regulatory review in Japan. But we have 28 antibodies in early stage pipeline. So I would say that this is a rather unique target. And also, interestingly, most of these antibodies have entered clinical studies after 2019. So it's also quite novel and apparently had a lot of success. And this is followed by HER2. HER2 has a totally different story. So the first antibody against HER2 has been approved in 1998, and there are 14 antibodies already in late stage regulatory review or approved. But still, we have 25 antibodies in early stage pipeline, which means that this is a very well validated target and a very interesting target. And as I will show you later, the R&D is engaging in also novel formats against this target. We also stratified our monospecific groups according to format subtypes in terms of conjugation or not. So most of the antibodies are naked antibodies, and 30% are antibody-drug conjugates, and then we have a 2% of radioimmunoconjugates. If we look at the targets in the first two groups, on the left you have naked antibodies in red, and on the right you have ADCs in yellow. And the striped bars refers to targets which also have an immunomodulatory property. So it's interesting to see how the targets for naked antibodies have mostly immunomodulatory properties, while this is not the case for the targets of antibody-drug conjugates. The most frequent one is HER2, followed by Claudin 18.2 and TROP2. And now we'll move to the second group of antibodies for cancer indication, which is multispecific and accounts for a third of the whole cancer indication early stage pipeline. Multispecific antibodies comprise bispecifics, trispecifics, tetraspecifics, but most of them are bispecifics, 88%. We have stratified this group according to the type of target. Most of these antibodies are cell engagers, followed by antibodies targeting the tumor only, and antibodies targeting immune cells only. And then we have antibodies targeting the tumor or the immune cells, but also the tumor microenvironments, such as cytokines. And I'm going to talk about in details about the first three groups. Cell engagers. These accounts for 66% of the multispecific groups. And as you know for sure, cell engagers are molecules which are able to engage on one side with tumor cells or fibroblasts, and on the other side with immune cells. We have stratified further this group in cell engagers only and cell engagers which also have immunomodulatory properties, which account for a third of the multispecific groups. And then we analyze the targets. Now you will see appearing on the left the popular targets for targeting the tumor, and on the right the popular targets for targeting immune cells. In the cells engagers only group, there is a variety of targets for targeting the tumor. The most frequent one is BCMA, followed by EGFR, CD20, CD19, HER2. While for targeting immune cells, most of the antibodies, 90%, use CD3. And the scenario is completely different in the group of cells engagers with immunomodulatory effect. So for targeting the tumor, the most popular target is PD-L1, and for targeting immune cells, the most popular target is 4-1BB. We do see CD3, but it's only in combination with immunomodulatory molecules such as 4-1BB or CD28. And now I move to the second group of multispecific antibodies, antibodies targeting the tumor only. This accounts for 17% of the multispecific group. We have done the same type of analysis. So we have stratified the molecules in antibodies only targeting the tumor and antibodies targeting the tumor, but also with immunomodulatory effect. And then we have analyzed the targets in the two groups. In the tumor-only group, the most frequent combination of targets is c-Met and EGFR, followed by HER2 only and c-Met only, but these are multispecific. So these are biparatopic antibodies targeting different epitopes on the same antigen. In the group with immunomodulatory effect, this scenario is different. So we have combinations of targets all involving CD47, and the most frequent one is PD-L1 and CD47. And as a reminder, CD47 is a molecule that if you block it, you abrogate the don't eat me signal. Going back to the in the tumor-only group, if we stratify by format subtype in terms of conjugation or not, we can see that a third of these molecules are antibody-drug conjugates. And this is very interesting because there's been lots of interest in the single format like ADCs or multispecific formats, especially in the last few years. And now we start to see a combinations of these two formats. So we have ADCs, which are multispecifics. Antibodies targeting immune cells only. These account for 10% of the multispecific groups. And if you look at the targets, most of them involve combination of PD-L1 with other molecules, such as CTLA-4, LAG-3, TIGIT, TIM-3, and this is to further fine-tune that immunomodulatory effect. And now I'm going to talk about the last group of antibodies for cancer indication, immunoconjugates, which are 8% of the antibodies for cancer indication group. We have stratified them according to the format, and today I'm going to talk to you about bispecific immunoconjugates, 46% of the immunoconjugates group, immunocytokines, 41% of the immunoconjugates group, and immunotoxins, which are only 7%. Bispecific immunoconjugates are bispecific molecules which have one antigen binding site that is derived from an antibody molecule, antibody gene. And the other antigen binding site is instead the fused protein. And here I'm showing you on the left the targets of the antibody portion of this molecule, and on the right is the corresponding fused protein. As you can see, the most popular target for bispecific immunoconjugates is PD-L1, and the most popular fused protein is TGF-beta receptor 2. And also you can see that the most popular combination is the PD-L1 and TGF-beta receptor 2. Immunocytokines. These there is a variety of targets for the antibody portion of these molecules. The most frequent one are PD-1 and PD-L1. And then here the fused protein is instead a cytokine. And the most frequent one is IL-15, followed by IL-2 variant and IL-12. And then we have immunotoxins. They are only 4, and they all target different targets. But the fused protein is mostly the Shiga-like toxin A. And now I move to the antibodies for non-cancer indications. We have stratified them according to the format, and 86% are monospecific, so the majority. We have only a 12% here of multispecifics and the 2% of immunoconjugates. A 3% of the monospecifics are antibody-drug conjugates, and this is 10 times less the proportion of antibody-drug conjugates in the cancer, in the antibody for cancer indication group. Then this is a very heterogeneous group, so we have stratified these molecules according to the primary therapeutic area. So almost half of these antibodies are for immune-mediated and inflammatory disorders, 46%, followed by infectious diseases, cardiovascular hemostasis, neurological disorders, ophthalmic disorders, metabolic disorders, respiratory disease, muscular disorders, skeletal disorders, and other. By other, we mean 2 molecules which couldn't really fit in any of the other categories, because one is for contraception and the other one is for methamphetamine intoxication. So this is to show you the variety of therapeutic area you can study and repurpose antibodies to. And what I'm going to do now is I'm going to give you some details about the first 2 groups and some of the other groups. Immune-mediated and inflammatory disorders. These account for 46% of the antibodies for non-cancer indication. And we have stratified them by mechanism of action. Most of these antibodies, 58%, act via blocking cytokine or chemokine or alarmin or TLR signaling. Then we have a 6% of antibodies with for immune cell depletion. 4% are checkpoint agonist. 3% block CD40 ligand pathway. A 2% block OX40 ligand pathway, and another 2% block immune cell recruitment to specific area of the body, for example, the brain, for the treatment of multiple sclerosis. Infectious disease. This accounts for 15% of the non-cancer group. And I'm providing you an overview on the target pathogens. Of note, we consider in this group only antibodies that directly target the pathogen or the host receptor for the pathogen. We do not include here antibodies which are meant to ameliorate the symptoms of the pathogen infection. These are mainly included in the immune-mediated inflammatory disorders group. So the most frequent target pathogen is HIV, followed by SARS-CoV-2 and HPV. And if you're wondering why there are still some antibodies against SARS-CoV-2 in early stage development, this is because they mostly they are meant for a specific target of population, which usually can't have the vaccine. Neurological disorders. These are 12% of the non-cancer group. We have stratified them by indication. Almost half are for Alzheimer's disease, and they mostly target amyloid beta and tau. A 14% is for Parkinson's disease, and they mostly target alpha-synuclein. And 11% is for pain, and they mostly target NGF. Ophthalmic diseases, these are only 4% of the non-cancer group, and they're interesting because here we find the majority of the bispecifics in this non-cancer group. So the most frequent target is a combination of VEGF and angiotensin II, and the most frequent targets are bispecific involving VEGF and other molecules. Muscular disorders. So these are only 2% of the non-cancer group and only 6 molecules. And if you are wondering why am I talking about this is because 5 of these molecules are actually a novel type of antibody. These are called antibody oligomers conjugates, and they are all targeting transferrin receptor 1, and they deliver oligomers such as siRNA, antisense oligomers, phosphorodiamidate morpholino oligomers, PMO, and these are specific for specific genes which are involved in specific muscular disorders. And now I will conclude with the overall trends in the clinical pipeline. With this slide, I'd like to show you the impressive increase in the numbers of antibodies in each phase of clinical development. Here we compare the number of antibodies in 2000 as of November 2018, dark green, and those as of November 2022, light green. And as you can clearly see, the number of antibodies in 2022 is more than twice the number that was 4 years before in phase 1, phase 2, and phase 3, which means that antibodies are nicely progressing to each phase of clinical development as well. Trends in first marketing approvals and success rates. With this slide, I'm showing you annual first approvals. And if you look at the green dotted line, which refers to all first approvals, you can clearly see a steady increase of approvals since 2010. If you look at the purple line, which refers to approvals which happened in US and EU first, and the yellow line, which refers to approvals which happened outside of US and EU, so in the rest of the world first, it's very interesting to see how since 2019 we start to see an increase of first approvals in the rest of the world. And this is very interesting. It means that the biopharmaceutical industry involved in antibody therapeutic development is growing more and more also outside of US and EU. And this is my last slide. And this is a preview on our latest analysis on phase transition and approval success rates. This will be featured in our next Antibodies to Watch in 2024, which will be published in MAPS by the end of this year. And here I'm showing you just a preview. And we calculated the phase transition and approval success rates of antibody therapeutics which entered clinical studies between 2000 and 2019. So this is a 20-year time period, which we divided in 10 years time periods overlapping. As you can see from the key on the right, we have antibodies which enter clinical studies between 2000 and 2009, between 2005 and 2014, and 2010 and 2019. And as you can see from the N numbers in brackets, the most recent the time period, the higher the number of antibodies, which is in line with the trends I showed you at the beginning of the presentation. And if we look at the antibodies which entered clinical studies in the 10-year time period after 2010, and we compare it with those which entered in the 10 years before 2010, except for phase 1 to 2, there is an increase in phase transition from phase 1 to phase 3 to marketing approval, and marketing approval to approval, which then translated in an increase of approval success rates, both globally from 20 to 30%, and in EU and US only from 19 to 28%. And these are my key messages. I hope I convinced you that the antibody therapeutics are entering clinical studies in increased numbers recently, especially those for cancer indications. Also, the biopharmaceutical industry is engaging in innovative R&D, and antibodies for cancer indications are mainly monospecifics, but a third are multispecific now. Most of the monospecifics are naked antibodies with immunomodulatory properties, and a third of the monospecifics are antibody-drug conjugates. Also, most of the multispecifics are cell engagers, and of these, a third have immunomodulatory properties. Of the antibodies for non-cancer indications, the majority are monospecifics, only 12% are multispecifics, and the majority are for immune-mediated and inflammatory disorders, but I showed you the huge variety of therapeutic area in this group. Also, trends show that antibodies are also progressing through each phase of clinical development, are being approved in increased numbers recently. And also from our analysis of success rates, phase transition and approval success rates, we can say that the antibodies which enter clinical studies after 2010, in the 10 years after 2010, compared to those who have an increased success rate compared to those who entered in the time period before. However, I wanted to mention it earlier, this group is the group with most uncertainty, because the proportion of molecules with unknown fate is the highest. We have a 40% of molecules which are still in active development. So as the fates of these molecules will be determined over time, this phase transition might change. And I would like to thank you, my colleague, Dr. Gianni Schreicher, Chief Operating Officer of the Antibody Society and Editor-in-Chief of MABS, and the Antibody Society and their corporate sponsors. Also, please join the Antibody Society to keep up to date. And if you're interested in the early stage pipeline, do consider becoming our corporate sponsor. We send our updated data set quarterly. Also, we are preparing the database, which will include all the data we have collected and we keep collecting on antibody therapeutics clinical pipeline, which will be available for our corporate sponsors. And now I'd like to thank you all our corporate sponsors, which are a lot. And I would like to thank you all for your attention, and I'm happy to answer any questions.
Excellent. Thank you very much, Silvia. And just to follow up on her thought, as she was going through the slides, if you happen to notice your company's logo there, your company, I'm pleased to inform you that your company has already paid for your membership. If you're not already registered as an Antibody Society member, please feel free to do that because your company has already paid for it. We do have some questions. So let's start with the one that likely popped up earliest, and that would be, can you elaborate on sources for all of this data? Where does it come from?
So we mainly rely on public disclosures from primary sources, which are mainly companies. So we go on the company website, we look at all the press releases, company presentations, annual reports. We look online for meeting abstract, articles, and also we look at what the companies upload on clinical trial registries, such as clinicaltrial.gov or others. And we also rely on other reliable sources such as regulatory agency documents, official documents, and also the WHO INN lists. But to clarify, we do not rely on commercial databases. We keep updating our own database.
Actually, I could add one more little thing into that. Silvia and I work together on that data set, which is enormous at this point. And I do want to remark that in the recent years, the past 4 or 5 years certainly, we have needed to be more proactive, shall I say, about going to the website of the Chinese regulatory agency and to track the clinical studies that are on Chinese clinical trials registries. So there's a lot of activity going on in China at the moment. And I would say over the course of the past 4 or 5 years-ish, the pipeline has gone from only having a few antibodies from Chinese companies to it being, I would say, about a third of antibodies that are coming from Chinese companies are comprise the pipeline. Well, 1/3 and then 2/3 everybody else. So tremendous amount of activity that we definitely try to keep track of, so you don't have to. Okay, next question. Next question would be, can you speculate on why there's a lower percentage of antibodies in the non-oncology area versus the oncology area?
So lately there's been a huge like an explosion of antibodies entering clinical studies for immune checkpoint inhibitors. I think since they appeared in the research and development field, and people started to see how successful they were, there's been a lot of interest and lots of people are working on them and also building up on them. So using them in different formats and finding novel immunomodulatory molecules, not only PD-1 and PD-L1, we have lots of them now. And I think that's one of the main things that happened when the two curves started to diverge.
I agree with you there. There's an awful lot of anti-PD-1s, anti-PD-L1s in that data set. Next question, which is right in your area of expertise, and that is, have you, or is there a way to, classify the therapeutic antibodies based on their FC biology?
Yes, of course. Yeah, we have, not for this analysis, but we collect all information about FC mutations and the reason for these mutations, and also the isotype, and also the different type of mutations and the effect that they have on FC effector function. So we collect all of these data, which are available for everyone for antibodies in late stage pipeline and antibodies which are approved. And you can go on our website. We have lovely searchable tables which you can use and look for all the information about antibodies in late stage studies and approved. We keep collecting this information also for antibodies in early stage, but this is not exhaustive because when molecules are in phase 1, sometimes there are not so many information available yet. So the more they progress into clinical development, the more information are disclosed. And so yes, we are very confident on our data on the late stage pipeline, but you will see this information also if you are a corporate sponsor in our early stage pipeline data set.
Thank you. One more quick question, and then we have a few about the success rates. The quick question would be, is this analysis focused on antibody formats only, or does it include alternative formats, DARPins, anticalins, etc.?
We consider, so for us, an antibody therapeutic is considered a molecules, which is a protein-based molecules, which has at least one antigen binding site that is derived from an antibody gene. And so whatever molecules which has an antigen binding site derived from an antibody gene of any format is considered. We do not consider anything else, things that are not this, that do not fall in this category.
And I failed to notice here that at the end of that, it does also mention single domain antibodies, so I can answer that myself. As that single binding site derived from an antibody gene, it is included, so single chain, variable fragments, single domain antibodies, those would be included as antibodies, but things like the anticalins or the DARPins are not. They're considered targeted protein therapeutics, but not defined specifically as an antibody. One more quick thing. Are you planning to include company name as one of the columns in the data set? So thanks for your interest in that data set. Absolutely. Of the company name as well as partners.
Yes.
So a couple of questions on the success rates. Let's see. Where's the first one? First one would be what contributed to the higher success rate for the more recent time period?
Yeah. So we have wondered the same. So very good question. And as you will see in our analysis that we show in our next antibodies to watch in 2024, we have further analyzed our data set, stratifying in antibodies for cancer or non-cancer indication. And what we saw, the difference we saw, is that the increase in the success rate in the time period after 2010 is mainly due of an increase in the success rate compared to the previous time period in the cancer group. And this again is we have done an analysis on the targets, and this appears to be mainly due to the checkpoint inhibitors. We had lots of success, so this is mainly due to that, according to our analysis.
Okay, next question on success rates, which you may have touched on it, but I'll ask the question anyway. How do you explain the decrease in phase 1 to phase 2 transition? Is it related to non-cancer indications or a specific mechanism of action class?
We looked into that, and I don't think there's a difference in the cancer and non-cancer indications group between in phase 1 to phase 2. But you will hear more in when we will publish the paper, and maybe we'll do more analysis on that.
And also bear in mind that, as Silvia mentioned earlier, the number of molecules for which we do not have a known fate is much larger in that larger time period. So I would not be terribly concerned about very relatively small-ish differences in the success rates between the different periods and comparing the more recent one to previous ones, because there's just a certain degree of uncertainty that we will not be able to address until we actually know what happens to those molecules. We are tracking the molecules in real time. We are watching what goes on every single day, believe me. I do. And the data set itself is over 10 years old. We really we've been at this for a while. The next question would be about success rates by format.
Yes, it's a very interesting question, and we are planning on preparing an article only based on success rate, where we will analyze success rates, stratifying our antibodies by formats, by targets, and do lots more analysis. Yes, it's a thing we are going to look into.
Next question again on success rates. I'm just reading through it kind of quickly. It's a longish one regarding the approval success rates of clinical trials. Is that due to the recent antibodies that are targeting highly verified molecules such as HER2, so well-validated targets like HER2, PD-1, or the antibodies targeting new molecules also showing an increase in success rates due to the improvement of understanding of the disease and signaling pathways? Probably a combination of both.
Exactly, but it's definitely a thing we are going to look into to answer this question, which is a very interesting question. But definitely when we did our target analysis for the antibodies to watch in 2024, the main targets responsible for this increasing success rate were actually PD-1, PD-L1, and HER2.
Next question. So let me just point out, there are many different ways of stratifying this data. We've taken a first pass at it. We are, in due time, going to do a standalone paper on success rates where we'll really delve into hopefully very fine detail. The next one is very fine detail. Will you consider adding what animals were used in immunization? There are many new antibodies from sources other than mice, and that is absolutely true. There's been a big uptick in antibodies coming from, or VHH antibodies, so coming from llamas, alpacas, but as well as rabbits, chickens, hamsters.
Yeah. We do have this information in our data set. We have the origin of the sequence. So, yeah, we keep track of that, and we might consider studying this for the success rate.
That is a very, very fine detail.
Yeah, very.
So it takes a while to dive into that, because I think, what was it, over 1,000 molecules in the early stage pipeline, and again, to emphasize that information about where they came from, how they are is commonly not known. So
exactly.
it can be quite difficult to try to do an analysis based on stratification for data where you only have part of the data. You can't really say too much about that particular variable.
Exactly.
And let me just take a quick look through, but I think we got to all of the questions. I'll give the going, gone talk. I don't see any more questions coming in. So thank you, Silvia, very much for
Thank you, Jan.
all of your work on the data set. It's incredibly time consuming to do this kind of work. Don't I know it? And this fabulous analysis, very well done, very well presented. I thank you very much for that.
Thank you, Jan.
And so, in concluding, I thank Dr. Crescioli for providing these insights and the trends in the early stage clinical pipeline of antibody therapeutics developed by companies. I also thank you for attending and being with us today for this webinar. An on-demand version will be available within a day. I will send a link to it via email to everyone who registered. So please do feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again and have a great rest of your day.